who have forgotten, I'm still Dr. Wyman. Still short, still bald, still charming. 2 out of 3 in that. One of the major advantages of working here is that I get to travel. Now, the travel part isn't because I'm on escape. I get to go out and I get to meet people and I get to take you all with me. So Rachel, Forrest, and I went to Honolulu. It was miserable. Yeah, I had my wife too. Awful. Just pain and suffering. I had to come back. And at the meeting, I met Alyssa sitting behind me, who's taller than me. And I helped her work on her resume. And within 5 minutes, I said, I'm going to invite her to a natural science seminar because she is smart. She is clever. And she's none of the things I am. So different one. She, I think, would be very impressive to you all. She is a fantastic young lady. She's working at Tom Sharkey's lab at Michigan State in the PRL. She is an honestly good biochemist. She actually does biochemistry. She does science communication and outreach. She wants to look at elements. Oh, not sure what's wrong with her. Feel free to ask questions when she's done. Feel free to come to lunch with us afterward. I think this is going to be a fantastic morning. I think you're going to really enjoy this presentation. She is going places. Watch her. Emulate her. Your faculty here, I know you love us. I know you can't get enough of us. But let's be honest, we're old. Her. And short her. And bold her. Other things her. Right? Feel free to ask her the things you won't ask us. She'll answer. You're on.
Great. Thank you so much for having me. It's really great to be able to come here and tell you a bit about what I do. So like you said, my name's Alyssa. I'll tell you a little bit about me, so I'm not just a person talking at you that you don't know anything about. I did my undergraduate and my graduate studies at Michigan State. I actually came in pre-med, decided to switch to plant biology, so now I study plants. And I started working in a lab as an undergraduate studying this transporter here, which I'll actually get to tell you more about because I ended up joining that same lab to do my graduate work and continuing that project. So I ended up graduating. They didn't let me out, but I came back. So I joined the Biomolecular Sciences program at Michigan State, which is where I'm at now. So the time that I'm allowed outside of the lab, I do a couple other things that I really enjoy. So this year I've gotten involved with Science Debate, which is a group where we talk to representatives. We're actually doing the gubernatorial races this year. We've mostly done the presidential candidates before. But we ask them questions about science policy because we think it's important that we know what they think about these issues that affect us. So we have a whole list of questions that they ask, and we make these publicly available. And then we talk about them, what we think on these topics. I've also been working with Plante, which is an online plant community. So I've been helping write articles and publish information with them. I've started this series. This is one of the things that I've worked on. I take scientists from different groups of plant science. You have the genetics, you have the ecology, you have the biochemists. We don't talk to each other very well, so I'm working on trying to bridge those gaps. So I do these infographics where I highlight a plant scientist in this category as we publish that so people can learn more about what other areas of plant science are doing. And then I'm actually also working on launching a podcast with another professor on campus. So what we're trying to do is take science that we know and that we're learning and make this available to the public because it affects things like what food you buy or what cleaners you get from the grocery store. So we want to help people make smart choices with science. So those are the things I get to do outside of my research. But I am going to talk to you about what I do because that's also really exciting for me. So first I want to spend some time talking about alternative pathways around the Calvin cycle. And first I'll tell you why that's important and why we're actually looking at this. And then we'll use those alternative pathways that we talk about to look at 2 different mutants that we have. We're working in Arabidopsis, which is a weed. It's a good model system, but it applies to a lot more plants. And so we'll apply it to these 2 problems that we've been looking at and how it gives us some answers. So first, we've been working on the Kelvin cycle for a really long time. You probably had to memorize it in high school at some point. They started work on this back in the 40s and 50s. These are some of the earliest ones that they drew out. They don't all agree. They aren't completely right. But we've been trying to figure out this puzzle for a really long time. And they got bits and pieces here and there, and eventually they got to this point, and this is the first published Kelvin cycle as we know it. But it kind of looks a mess like this. So now we redraw it like this. So this was figured out back in the 50s. So what you have is plants take up CO2. They use light energy during the day. They make PGA, which is 3 carbons. You take 5 carbons plus 1 carbon, split it in half. And you can make these triose phosphates. And these can come out of the chloroplast where they're originally made, and you can use that to make sucrose. And that's what the plant uses during the day for all of its energy and growth. It's immediate fuel usage. Or you can keep it in the chloroplast, and you can get starch, which is what it used to survive the night. So it's kind of like us. We need to always be consuming energy, so this is how plants make sure they can space it out through the entire day. And so this has been discovered in the 50s, and a lot of work kind of stopped at that point. We know the cycle, we know how plants do this. There's not too much more to figure out. But what's actually the case is that it's a lot more complicated than just this. Each of these arrows is an enzyme that's doing a reaction. They're all regulated, they're all interacting with each other. These two pathways interact with each other. You have to make sure you get the right amount going to each side. And so there's a lot more questions to be addressed in this. And so that's where I kind of picked up this question. What do we not know about this very basic thing with plants? And what can we figure out? And one of the reasons I got really curious about this is because I started to realize how important plants were. So I said I went into undergraduate going pre-med, and I eventually switched to plant biology. That's because I joined a plant lab for a summer. And I wasn't planning on staying there, but as I got to know the project, I saw how essential plants were. And I don't think it's an exaggeration to say we wouldn't be here without plants. We need them for food, we need them for energy, we use them for medicine. And so I saw more and more how plants affected everything that we do, and this is the core thing that plants do. I mean, if you ask someone, what do you know about plants? Probably say something like they use sunlight to make energy. And so we want to understand the basics of this thing that we're all so dependent on. So I'm going to tell you about 3 little stories, and we'll focus in on one of them about these alternative pathways that we're looking at. So carbon doesn't always take this route. It does most of the time, but there's other things plants can do. So one of these is what we call the Rubisco Shunt. So in seeds, you take pyruvate and you make triglycerides, and that creates a lot of oil in the seeds that you can use to power them as they start growing. But when you do this, you give off carbon dioxide, and so you're losing one of your precious carbon molecules that you could use for growth. So what seeds do is they add Rubisco into seeds. So normally, that's the first step of the Kelvin cycle. You add CO2, you make that PGA that I was talking about, and eventually it cycles back and you just keep doing this loop. But then you get stuck if you just keep doing a cycle. You're not using it for anything, you're just tying it up somewhere else. So what they've figured out how to do is you take out this extra loop, you make it into pyruvate, which is also the first step over here. And so you create this cycle instead. So eventually it's more productive because you can take all that carbon you're losing and you can stick it into something that's actually useful. So plants have come up with these kind of shortcuts to make things more efficient. And one of the ones I've been working on for my project is what we're calling the glucose 6-phosphate shunt, which is just a big word for this molecule right here. I'm going to call it G6P. It's a sugar that plants use in both starch and sucrose synthesis. So this is what it looks like. You take glucose 6-phosphate, you bring it around, and eventually you get to your pentose phosphates, but you lose a carbon dioxide in the process. And so this doesn't seem like it's extremely beneficial, like we talked about. You're losing one of those carbon molecules that you could be using for growth or defense or anything else you need your plant to do. But we think there's some cases where this actually could be a useful thing. It can help stabilize things and it can help regulate things, which I'll be getting into. So what we have here is that carbon comes in, carbon comes out. You use 2NADPH right here in this energy step. It's one of the things that electron transport makes, so you can do other things in the cell. But you also make 2NADPH down here. So these two cancel out. You use 2, but then you make 2. And then you also use 3 ATP, which is another form of energy. So you ultimately lose 3 ATP, but you don't lose NADPH because you just make it again at the end. So I'm going to walk through what this means and why we think it could be beneficial to plants to have this extra pathway. So we're going to focus this on this molecule here. This is what G6P looks like. So this is what the pathway is named for. It's this starting molecule that you have to begin at. And so you can make this with fructose 6-phosphate, it's another sugar, and this enzyme, PGI, is what converts these two back and forth. You can take it and you can use it to make starch, like we talked about, the overnight energy. You can take it this way with G6PDH, which this is the beginning step of the shunt. It's the first committed pathway, so once you do this, you're stuck and you can't go back. Or there's a transporter that can bring it inside and outside of the chloroplast and bring it back and forth. Now this usually doesn't happen in plants. There's some cases where it can happen, and I'll talk to you about those, but it's not usually up. If you just grab a leaf, it won't be doing this. So first I'm going to focus on this enzyme right here, G6PDH, which is short for glucose 6-phosphate dehydrogenase. And so it just does this reaction, but this is what I've spent a lot of my time working on because it's the first step and it's the first committed step. So once you do this, you can't go backwards. So it's really important to figure out how it's regulated and why and when it does what it does. So a lot of work was done on this back in the 80s, so I've spent a lot of time going back 20 years in papers and rereading all these papers that were like hand-drawn graphs. So this is one of those. So G6PDH is thought to be off during the day. It's redox regulated, so when sunlight's hitting it, it changes the redox status of the cell, which makes this go off. If you put it back in the dark, the activity goes back up. So you get it off during the day to about 20%. And what we saw is if you have these enzymes, there's 3 different types in the chloroplast, which I'm just going to call 1, 2, and 3. So the first one, and this is what's thought to be majority of what's in the chloroplast, it's the one that's redox regulated. It's causing this downshift in activation. So it goes from 100% activity all the way down to about 20%. The other ones stay at about 80%. So we saw, yep, what they saw before is still happening. That seems pretty simple. It's off during the day. But I wanted to understand how it was turning off. And so I did, sorry. Slides always really slow. So I did these kind of experiments. So I don't know if you've done enzyme assays before, but really quickly, what you do is you give it the substrate, the thing that it's going to be working on, and you increase the concentration, and that changes how fast it goes. So very typically, it comes up very quickly, and usually it'll plateau up here. And what's interesting about this is we don't see a plateau like you would expect, but it dips back down. So if you add too much substrate, it starts clogging up the enzyme, and you get inhibition. And then we also saw, if you're looking at the difference between the red and the black, the black is the on state, the red is the off state. And so what you have is not a shift in in Kcat, but you have a shift in Km. What that means is Kcat is kind of like the gas pedal of your enzyme. You can speed it up or slow down, but the whole thing goes up or down together. That's not what happens. What we see instead is more like one of these climbing walls. So if you have a really tall wall, you can still get over it. You just need a bit more of a running start to get over, and you need a bit more oomph to get over it, but you can still get over it. So what we saw is not a brake pedal, but we saw just a bigger wall that you had to climb over. So you could still get the same amount of activity, you just needed to give it more substrate to work with. So that was interesting to us, because if you get conditions where you're getting more substrate in the chloroplast, you can still get a ton of activity from this thing. It doesn't turn it off in the way that we would expect. One of the other things I looked at is how it's actually regulated, how you turn it on and off. So that's through redox regulation, which I won't go through all of this, but pretty much you take these cysteine residues and you can connect them together. So a lot of enzymes use this to either kind of put a lock over the enzyme, over the active site. So if you have your active site open and then you link these two over top, nothing can get in, and so it turns it off. This is working a different way. I'm still working on this, but what I think it's doing is that it's kind of loose and untangled if they're not linked together. When you bring them together, it makes sure that the enzyme can actually work and it's folded properly. So it's redox regulated, and the way that you do this is you can pass these little bridges from one thing to the other. So it can go from oxidized to reduced, back to oxidized, but as it does that, it passes it on to another enzyme, which can keep on going down the chain. And so you can compare all these, and you can see how similar they are and how easy it is to pass these little bridges on from one to another. And you get something like this graph. You give it a certain potential, and that lets you make or unmake these little bridges. And if you do the analysis of this, you get a whole series of numbers, which don't mean too much. It's useful to compare, but only if you know all the math and equations that go behind it. But what it tells us is we can come up with these predictions. So these are a bunch of enzymes that are normally working in the Kelvin cycle in electron transport. So they need to be fully on during the day. You don't want to have those off at all. You need them to be going as fast as you can. And so if we set these to 100%, we can calculate that this is only at 50%. So it's not all the way off, it's only halfway off. And so even already we're seeing a picture that it's regulated differently, and it's more on than what we think it is. So it's probably getting a lot more activity than we had previously expected. And what this is kind of looking like in my mind, a lot of people used to think it was kind of regulated like this, either on or off, one or the other. The picture that I'm starting to see is more like a dimmer switch, where you can change it based on what you need, and it's easily manipulatable. You can have it all the way on, you can have a little bit, and you can change it throughout the day based on what your plant needs. And so I wanted to look at plants and see if this was actually happening. It's all well and good to take purified proteins in a test tube and make them do what I want, but is this actually happening? Does it actually matter? And so what I saw is that if I look at my G6PDH in the plant, it's more active than what I was seeing just in my purified stuff. So if this is the predominant enzyme, G6PDH1, if this is what's controlling most of the on and off activity, you'd expect in the plant for it to go down to about 20% when it was all the way off. But this isn't what we see. We go from 1 or 100% down to about 50, 60% in the plant. So this is confirming what we're seeing already, that there's more activity than what we think. It's more regulated than what we think. And so the big picture that we come away with is something like this. There's two ways to work with this enzyme and to increase the activity. Like I said, you can increase the G6P, get over that higher wall. So you go from here up to here, but you get more activity. Or you can change the dimmer switch of the enzyme. So you can go from here up to here, and you also get an increase in activity. And so there's different conditions in the plant that can cause both of these two things, but it hasn't been considered before. It's thought that it's off, and so it's not important. So what we're showing is a story that this is important, this is most likely happening, and it's something that we need to consider when we're thinking about photosynthesis. So next I want to tell you a little bit about this enzyme here, GPT2. So this is a transporter across the chloroplast membrane. So these are the same molecule, they're just moving from one location to another. And so what we saw is that you can get this if you block starch synthesis. So if you take these enzymes A, B, and C here, you can knock them out so the plant doesn't have them anymore. So normally it would take F6P and it would make starch to use overnight, but we stopped that from happening. And then what the plants do is they increase this transporter here. So it brings in this sugar from outside the chloroplast. So we were wondering, why is this happening? It's interesting, but why is the plant doing this? And what we think is that, especially for this first one here, it helps bring it in this glucose 6-phosphate. And then you can still make some starch as long as you have these two enzymes. You don't get nearly as much if you knock these out, but it helps the plant recover and still keep surviving and not starve throughout the night. And we tested this in a couple different mutants in a different couple of conditions. So PGM is one of those knockouts, and we saw that transcripts, the message that makes this protein, does go up. And then we also saw if you put the plants in a high light, we can also make them do this. So now we have two conditions where we know we can get this sugar across the membrane. If you knock out starch, and if you put plants in high light. There's also another study done by a different lab that showed if you grow plants at high CO2, this happens as well. So it's this one down here, and it switches from this light blue, which is not much, to this dark blue, which is a lot more transcript. So it has this huge change in transcript under three conditions now: block in starch synthesis, high light, and high CO2. So we know that there are conditions where the sugar is crossing the membrane. And so we wanted to think about what are the implications of this? What does it mean for the plant? And so we came up with this idea of what we're calling the cytosolic bypass. And there's a lot more to that, but that part's not as important. So what happens is you can bring these triphosphate out and start making sucrose. But because they share a common molecule, you can start transporting them across. Which leads into another protein that I was studying, which is this one up here, PGI. So if you go back, that's this one right here. So if you're bringing in G6P, it has a couple different pathways it can take. And it could take PGM, G6PH, or it could go backwards to PGI. This is reversible. And so I wanted to look at how reversible is this? Can it actually go backwards? And what we found is that it can't really, at least not very well. And so if you look at the measurements of F6P and G6P, these two, in these two locations, they're quite different. You would expect something that can go back and forwards really easily to eventually come to an equilibrium where you have the same amount of both. but that's not what happens. This plant purposely stays at disequilibrium. And so you see a ratio of about 3 to 4 out here. And that's why if you calculate it and you do it in the test tube, this is what you would expect. But in the chloroplast over here, we see a totally different ratio. And this is not what we'd expect the protein to be doing. And so it's purposely keeping it at disequilibrium for some reason to regulate the amount of glucose 6-phosphate and fructose 6-phosphate. And the way that it's doing this is a difference in Km. And so that's that wall that I was talking about. In the plastid, where we see it come to what we had expected to, they have the same Km for both F6P and G6P, the two substrates that are going backwards and forwards. And so that makes sense. If it's coming to equilibrium where we would expect, they have the same Km. But the reason that it doesn't in here is because there's a huge difference in Km, over twofold. And so the mechanisms of how the protein is regulating that height of the wall is keeping this protein from coming to equilibrium. And we think that's because if you get too much of this glucose 6-phosphate, you're giving it more energy to get over that wall, and you could start doing the shunt in conditions where you don't want it to happen. So you want to keep this amount as low as you can unless you really, really need it. So I want to use those two last pathways we talked about, the cytosolic bypass and the shunt, to explain two mutants that we've been looking at. So we have these ideas. We want to see if the plant's actually using them and if they can explain things we've been seeing in the real world. So what we see is that, like I was saying, G6D can activate the shunt. So apply it first to this HPR unit. It doesn't really matter what HPR stands for. But when you do photosynthesis, you can either take carbon dioxide and you can stick it on to that RUBP and then go forward. But because carbon dioxide and oxygen look quite similar, and there's a lot of oxygen floating around in the air, you can also stick an oxygen onto that. And it gets you stuff that isn't useful for your plant. And so you need something to do with it to get it back to being useful. And so this is the pathway that takes. So you take RUBP plus oxygen, and you go through this whole series of reactions to get it back to something that you can use again. And one of these enzymes is HPR1. So what happens is we have knocked this out in one of our plants, and it does some funny things to the plants. They look really small, really sick. They don't get much bigger than that at all. And so there's something funny going on with them. There's a reason that they need this enzyme. And so we've used these hypotheses to figure out what we think is happening in these plants. So in these plants, 2PG, which is just another molecule, it's the product that comes right before this, 2PG accumulates. So you get higher amounts in these two lines of this mutant. And what that molecule does is it inhibits an enzyme that you need for photosynthesis. So it stops this reaction from GAP to DHAP. And so you can't get any of the stuff that you would need to do photosynthesis. You get stuck because the enzyme's inhibited. And so it makes sense that the plants are sick because they can't photosynthesize properly. You're stuck. You can't get any more sugar that's useful. So what happens then is this first molecule right here accumulates. You get a lot of gap right here. And so when you get that, you have concentration gradients that build up. And so you start taking that outside, just because there's so much of it, it has to go somewhere. So you start bringing it out, and then you can make sucrose again, so you're happier during the day. This is looking a little bit better for you. But then what we see is that if you put these plants in high light, especially, GPT2 goes up. It's that protein that we're talking about, that transporter. So now you can bring GAP out, you can make sucrose, but then you also start bringing it in, and you can start making starch again. But because you're bringing it in, you also build up this concentration gradient, because it doesn't like to go back to F6P. That's what we were talking about with PGI. It gets stuck at G6P, it doesn't like to go backwards. So some of it does, and you can start getting around this way, but not super easily. So a lot of it goes to starch, but some of it goes this way, into the shunt. And so you can keep turning it over, and that's why you actually have plants that are alive and can grow, because you can keep fixing carbon, you can survive the nights again. So they're not happy, but they can survive, and we think it's because they're doing this right here. It also explains some of the other measurements that other labs have seen. So the shunt helps explain one of these measurements that we've seen. Here you have this blue line, and that's normal photorespiratory CO2 that comes out. Plants normally do this, they give off CO2, and so we'd expect to see some amount of CO2 coming off. But in these HPR plants, these white dots, you see a lot more CO2 coming off than what you would expect, which is coming from the shunt. It gives off the CO2. So it makes sense that in plants that we would expect to see more activity through the shunt, we would see an increase of CO2. So we're going to switch to another mutant now. So we worked through HPR. We're going to now look at HCEF, which is a mutant that cuts it off right here. So it gets rid of this enzyme. So again, you're running into the same problem. You can't keep turning over the cycle, but you can at least do this. So what we think is happening is very similar to the other one. Carbon comes out, starts making sucrose, comes back in, and you start doing the shunt, just like the other ones. So we want to think about what are some of the other things that we know about this mutant? How is it working? So one of the things that this mutant was actually interesting, it wasn't identified because they were looking for a knockout of that one protein. They were looking for something else and happened to find this one. And so they were looking for something called cyclic electron flow. So if you're doing electron transport, you're taking energy from the sun, you're making useful energy for the plant. This is one of the things you can do. So what happens is you get your electrons coming from PS2, you eventually pass them to PS1 and go to NADP to make NADPH. That's what we talked about using the Kelvin cycle. It's how it keeps making more carbon. But what you can do is you can take that energy and you can pass it back. So it keeps doing this little loop right here, and it never makes it all the way to the end. Doesn't seem particularly useful, but what it does do is it keeps pumping these protons across. And what that does is it helps make ATP. So you can make ATP, but you don't make NADPH. And that's how they identified this mutant, and eventually they figured out why it was a mutant, but this is how they first found it. So this is starting to sound quite similar to our shunt. Cyclic electron flow makes ATP, but it doesn't make NADPH. The shunt consumes ATP, but it doesn't consume NADPH. So we're already seeing some connections between electron transport and carbon. And this is one of the big questions that's still remaining in this field. You have things like electron transport, carbon fixation, and end product synthesis, making that sucrose and starch. And all these have to work together because they're all heading to one goal. So you can't have one go faster than the other. If your electron transport was going a lot faster than what you can make at the end, you're building up a lot of useless energy that you can't do anything with. But if your end product synthesis is going a lot faster than you can make energy, you're not going to be able to make enough. So they all have to work in tandem, but we still don't really know how they're doing that. So we're seeing that there's some ties between carbon fixation and electron transport, and this is one of the first times that we're trying to figure out the connection between these two fields. So this allows the plant, okay. So this allows the plant to keep the ratios of the ATP, the NADPH similar, while still doing this cyclic electron flow, which can be really important. It helps the plant protect itself, especially under really stressful conditions. And so if there's a lot of stress, you want to be able to do that, and the shunt allows it to continue happening to keep protecting the plant in the way that it's supposed to. And so we looked at one of these mutants. I was telling you about GPT-2. If we get a lot of GPT-2, we start doing shunt. And so we looked at an overexpressor, which just makes tons and tons of it, and we do see cyclic go up. So this is a bit confusing of a graph. But what it means is that for a given amount of protons going across the membrane, so amount of protons that you're pumping, how much linear electron flow you're making. So how much eventually goes to NADPH. And so in wild type, you would expect, say at 4, look down here, this is a bit more obvious. So at 4, you would expect about 20 to go to, or 20% to go to linear electron flow. But with the overexpressor, you see a lot less, which means that energy has to be going somewhere else, which is cyclic. So it's a bit confusing. You have to do this measurement kind of backwards to get at what you're getting at, but our collaborators do a really great job at getting these difficult measurements. And so we're starting to see that if you activate the shunt, you can get cyclic. These two are seeming to be tied together. And one of the other interesting things that I've been working at recently is they're regulated by the same thing. So if you take plants and you give them hydrogen peroxide, this is your cyclic electron flow on this axis and amount of hydrogen peroxide, you get increased cyclic with the amount of hydrogen peroxide that you add. You also see this in that mutant that I was telling you about. They have increased hydrogen peroxide. And so I wanted to know if our protein that controls the flux through the shunt was also regulated by hydrogen peroxide. And it turns out it is. So you can recover, if it's off, you can recover almost all that activity if you give it hydrogen peroxide. And so we're starting to see that these are regulated by the same thing. They seem to be doing similar things and provide this connection between these two components of photosynthesis that we didn't really know how they were interacting. And so this is where I'm really interested in going in the future. I want to know how these three things are talking to each other, how they interact, how they regulate, how they work together. And that's one of the big areas that's still remaining in this field. We know each of them individually or are getting to know them pretty well, but it's these points in between that are still missing, and that's where I'm hoping to fill in some puzzle pieces. So just to summarize, we talked about some alternative pathways, different ways that plants do photosynthesis, especially the Rubisco Shunt, our new hypotheses of the G6P shunt and the cytosol bypass, and then how that explains some of the previous mutants that we've been looking at and gives us some answers for why they're working the way that they are. So I'd like to thank my lab, especially Sean and Tom. These are my two mentors in the lab. They've done a great job getting me through so far, as well as Abrigida and Evan, two other people in the lab who were working with me on this. I'd also like to thank the WHO lab and the Kramer lab. They did the cyclic electron flow work and some of our other mutants analysis. They've been a huge help as well. And then our funding. Thank you, and I'll take any questions.
As you formulate your questions, a couple things I want to point out. You'll notice she talked about electrochemistry and redox chemistry, which you learned about in Chem 111 and Chem 112. She talked about a number of functional groups, including lactones, that you learned about in Chem Organic 1, Organic 2. She talked about photosynthetic pathways, which you've seen in Bio 112. KM, MAX, KCAT, you will see in biochemistry and everywhere else. You know this stuff. Right now, for many of you, it's a set of disparate pieces. I assure you, keep practicing. It comes together. Everything we show you, teach you, have you delve into, set on fire, if you're Dr. Councilman, it all comes back together. It's going to be okay. There's a lot of panic in some cases. There's some of you like, all this stuff, what do I do with it? It's going to be okay. Have faith in yourself. Keep practicing. Now, I know several of you have questions because I suggested you should have questions. So the plants can take it, planted people can work in a minute, but if it isn't planted, I use that pun intentionally. I don't know if you're not. Anyone, questions for Alyssa? Go ahead. Go.
I have a question about the hydrogen peroxide regulation. So does the radicalization of hydrogen peroxide affect the plant? Like, is that dangerous for it, or is it safe enough?
Yeah, so normally when you have hydrogen peroxide, it's a balance of how much you have. So it can be really dangerous for the plant. And if you build up too much of it, it does start destroying things and kind of going crazy, especially the lipids. It tends to tear those apart really easily. But plants also use it as a stress response. And so plants do make it purposefully for stress because it starts all these other signaling pathways. So you want a little bit of it when you need, but you don't want too much of it.
Can you speak a little bit more to what made you change from more of a medical focus in school to a plant biology?
Yeah, it was kind of just like random chance, honestly. So I needed a summer job, and Tom hired me, and so I started working there. I thought it was just going to be for the summer. But I started working on the project of the GPT-2 transporter. So they had me reading all this stuff on photosynthesis and why this is important. I had to do presentations. So I needed introduction slides that talked about why we do what we do. And as I started thinking about that, I got really interested in how much of our life revolves around plants and how much they do. And it's really quite incredible that they're able to take energy and air, like sunlight and air, and do all this stuff. So like if you look at the redwoods in California, they're absolutely huge. They're amazing. And all that came from the air. Like it's just incredible how they work. So I started to get more and more excited about plants, and eventually I'm hooked. I'll keep doing this.
What kind of instrumentation did you use to collect the data, or was used to collect the data?
Yeah, so we use a couple different things. A lot of what I spend my time doing is enzyme assays, and so I use a spectrophotometer, which measures absorbance changes. NADP, NADPH is really nice to work with because it has a really distinct absorbance change at a very specific wavelength. And so I give my protein the stuff, and I watch the absorbance change, and then I can measure the slopes and the differences, and that gives me most of the data that you saw. We use other things to measure fluorescence. We have a little machine that you can clip on plants, and it measures fluorescence and how much CO2 and O2 there is, so we can figure out things with.
Are those the Kramer units?
We do a lot of stuff with them. It's LiCor, but we have a lot of custom stuff from the Kramer lab too. Yeah, we work a lot with them. So then we can measure things like how much the plant is photosynthesizing, what it's actually doing in the leaf too. So I didn't have as much of that data today, but we do a lot of that as well.
So those who have had Chem 111 and 112, you've played with a fair amount of handheld equipment. Imagine taking that to the next level. That's what she's talking about. A device, a smartphone about yay big, that you can scan across the plant. It can give you 20 unique data points immediately. Instrumentation that 20 years ago, when single instruments would have cost several $1,000s a piece, you can now get for $499.99.
It's really cool because
And
a lot
all
of that,
wireless.
yeah, a lot of that electron transport stuff that I was telling you about, you can look at those individual components and figure out what they're specifically doing with those machines, which is stuff that seems very abstract to me, but they can do it. Yeah.
Did you mention you measure CO2?
I don't, at least for this project right now, I'm getting into that in my next set of experiments. We have a lot of other members in the lab who are doing photosynthesis, photo respiration measurements.
Is that done fairly simply, or is that?
No, it's quite easy to do. We have this machine called a LiCor that you just clip on it and it starts measuring it.
Is it voltammetry or is it?
It's infrared.
It's infrared. I can show you the website now.
How much what
Couple $100
dollar signs you're talking
Couple
about?
$100.
Really?
Yep. I can get a deal.
Really?
Yeah, I just have it.
Take an offering?
Pass the plate, see what happens.
They're really fun. There's a lot of really cool stuff that we're doing. So next set of experiments I'm hoping to is take different mutants and see how much CO2 they're giving off, how much photorespiration they're doing compared to other plants. So I'll be getting to that eventually.
Great. What about oxygen?
Same thing.
Same thing?
Same machine does it.
Okay.
Sure.
So one of the reasons Alyssa and I hit up pretty quickly, besides the fact that we're back to the same person,
is the fact that she's in photosynthesis, and Tom and I did our work in the same lab at U of M in the photosynthetic lab. And we worked on this complex right here, photosystem 2. We looked at how do photosynthetic organisms take water, the most stable thing possible, and split it intentionally for electrons. Now that should be the simplest thing ever, right? You know, why would you attack something inherently stable? She works up from the other end. Electrons have already been free. Where are they going? And it's a fascinating piece. She's not kidding. This system's been studied for over 100 years. We know this much. Wow. And it's a room-sized question. Don't think that science has been shown. She kept saying that over and over again.
Yeah, I remember that. Yeah.
What she didn't tell you is that when Calvin was doing these assays, you're free told the Calvin stories.
Oh yeah. We get told that story a lot.
Yeah. He would mouth pipette radioactive isotope.
Bad choice.
He died of throat, mouth, and tongue cancer.
I think their lab is still radioactive.
It is so radioactive, I understand. Yeah. Yeah. It's the thing with that, mouth pipette, that's your fright to start with, and then mouth pipette radioactive compounds, tracers. Fascinating system. It's this idea of, okay. So, and that was data that sat there for 25, 35, 45 years until the Sharkey lab picked it back up, because it was proven gospel. Science moves. Science progresses. Okay? Never presume something is established. And don't be afraid to say, no. It looks like an orange, but I'm not sure it's an orange. If you all have more questions, you're free to stick around and ask them. You've been a great audience. That was a fantastic talk.
Yes.